Thank you, Art, and it's really my privilege to be able to introduce Brendan Harley today. Brendan received a BS in Engineering Sciences from Harvard University in 2000 and a doctorate in Mechanical Engineering from MIT in 2006. He was a postdoctoral fellow in the Joint Program for Transfusion Medicine at Children's Hospital Boston and Harvard Medical School from 2006 to 2008, and in 2008, he joined the faculty at the University of Illinois at Urbana-Champaign, where he's assistant professor of chemical and biomolecular engineering and directs the Engineered Cellular Microenvironments and Microstructures Laboratory. There's quite a lot that I could say about Brendan because I think he really is one of the rising stars in our field. But I want to clear the stage relatively quickly so that he can tell you about some of his wonderful recent work. But I do want to say a few things. First, about Brendan's approach to biomaterials research. Brendan's interdisciplinary training has led to an appreciation for the complexity of tissue engineering and the need for multifunctional biomaterials. In a scientific environment in the United States that's been recently criticized for promoting risk-averse mindsets among young investigators, Brendan's been willing to take on high-risk, transformative research topics. As a result, he's made remarkable progress toward addressing some of the most challenging questions in our field, such as how is tissue organization controlled at tissue interfaces, and how can the characteristics of native stem cell niches be recreated ex vivo. His goals are ambitious, but addressed with outstanding scientific rigor. A few recent examples include benzophenone-based protein patterning in 3D scaffolds, including growth factor patterning, composite scaffolds with controlled physical and mechanical gradients, and periodically perforated core shell biomaterials as 3D scaffolds for tissue engineering. Just these three examples remarkably demonstrate Brendan's ability to innovate in materials chemistry, mechanics, and structural organization. Importantly, Brendan's also creatively combining these distinct biomaterial properties in high throughput systems in order to optimize tissue regeneration. Brendan also combines his diverse scientific training with resourcefulness. And I'll tell one story from a personal perspective. A few years ago, Brendan saw one of my talks in which I was describing some molecules that we had engineered. And Brendan became interested. We had a bit of a discussion afterwards. Immediately after, I got an email from Brendan requesting some of these molecules to include in one of his scaffolds. We didn't hear very much about it for the next 6 months, and then he sent me a manuscript draft that included in some portion some of the molecules that we had provided and some of the really innovative things he had done with them. So obviously, Brendan's laboratory is moving quite quickly and really has quite impressive capabilities. This is really just one example to demonstrate the innovation, resourcefulness, and impact of Brendan's work. And that paper, I guess, as a side note, was provisionally accepted yesterday in a prominent materials journal. I suspect others in the audience may have similar stories about Brendan's interdisciplinary innovation. I also want to talk about Brendan's leadership potential. In addition to his outstanding scientific resume, Brendan has shown unique potential for leadership in our society. He serves as chair for the Engineering Cells and Their Microenvironment SIG in the Society for Biomaterials, previously served as vice chair of the Tissue Engineering Special Interest Group in the SFB. He has served as an editor in the Journal of Mechanical Behavior of Biomedical Materials and co-authored the book Cellular Materials in Nature and Medicine. Anyone who's ever met Brendan knows that he has a uniquely collegial mindset that's conducive to building partnerships and really enriching our community. Brendan's work's already begun to make a significant clinical impact as he co-founded a company called Orthomimetics that was recently acquired by Tygenix, and that company is currently performing phase 1 clinical trials on a biomaterial Brendan helped develop for osteochondral repair. Brendan's won numerous awards already for a diverse set of accomplishments, including the NSF Career Award in 2013, the Everett Award for Teaching Excellence at the University of Illinois College of Engineering, and today he joins a distinguished list of previous winners of the SFB Young Investigator Award. Brendan's trajectory is clear and suggests this will be the first but not the last time he speaks at one of our plenary sessions. So without further ado, it's my privilege to have Brendan as a friend and a colleague, and my great pleasure to present the 2014 Young Investigator Award to Brendan Harley.
Well, wow. So, Thank you, Bill. I very much appreciate the opportunity to come up here and represent the excellent work that my students have been doing over the last few years. And so I want to start with a few acknowledgments. And first, Bill's wonderful mug up there on the screen. Bill, I'm not quite sure how we met, but many years ago he took an interest in my career, and it has been a transformative experience, to say the least, to see someone who I want to model my career after. And so it was a great amount of pleasure I got to have Bill nominate me for this award, and my letter writers who supported me for this award, but also the community in general. Dr. Mipkos and Pepos, the president and the chair of the Award and Ceremonies Committee. I also want to make sure to thank Dan Lemaire and Leslie Clark and the rest of the Society staff for making this program run, and congratulate the other award winners this year, as well as everyone who was part of this community. I want to paraphrase something that Dr. Bizio said on Wednesday, is that this has really been a welcoming community for me, and I very much appreciate the opportunity to speak in front of you. My first experience in Society for Biomaterials was 2002 back in Tampa, and I was excited to be giving a talk, the first talk I ever gave at a scientific conference on degradation rates of collagen tubes for nerve regeneration. And I got up and gave my talk, and right after me, Dr. Ravi Belankanda, who's still at Case, got up and gave his talk. And the thing that struck me is that we spent 10 minutes, I don't think he probably remembers, but we spent 10 minutes after the session talking about my work and his work, and that was the welcome to the community here. And since then I followed up with coming to almost every meeting I can, and it's just been a wonderful opportunity for me to be part of this community, so I'm truly humbled to be part of it. One last thing I wanted to mention to the junior trainees out there is it took me until 2009 to get involved in the SIGs, and I wish I had done it earlier. It's a really unique opportunity for you to build your leadership and to build your connections within the society. And so I also want to make a few other acknowledgments from the beginning. My family. And I promise I won't become too emotional on this, but it really is a representation of us as a family unit that makes this possible. My daughter Joan, who, despite her love of pirates, swashbuckling is not her favorite word. In fact, it's scleraxis. She likes coming to my office and pretending to be a scientist and writing scleraxis on the board. And my wife Kate, her research is an anthropologist studying the evolutionary context of the endometrium, has been, a great person to share my relationship with, but also her work running the Safe 13 program, which is a program she initiated to document sexual harassment and sexual assault at field research sites around the world that's rampant amongst a number of other academic disciplines, is a huge risk that she took early on. And so those two have taught me that it's important to do what you love, with who you love, and to do it in a way that honors their commitment to you. And so I very much appreciate my family to make this possible, as well as my extended family. So I've had a wonderful group of trainees that have worked in my group. Stephen and Sonny and Nate and Emily and Bill and Rebecca and Bhushan and Laura and Sarah and Jackie and Dan, as long as our two newly regenerating members. That's the best job of regeneration we've done in the lab so far with Alonzo and Mara. It's a truly wonderful opportunity for me to be able to represent their hard work up here, and I'm very much appreciative for what that they did to make this possible. And lastly, I want to thank my mentors. I've had too many mentors to list. In fact, I tried to put a slide together of the faces of those people who impacted me professionally through this society, and it became a page that, even though the screen is large, the faces would have been incredibly small. And so there's too many of you to mention, but you have all contributed to my my acceptance into this society, and it is truly meaningful. But I do want to mention three people: Yanni Anas and Lorna Gibson. They were my advisors in graduate school, and the two of them gave me a really unique insight into the field of tissue engineering. Yanni from his early work with collagen biomaterials and the idea of regenerative medicine, where he was always pushing me to think about clinical translation and to think about regulatory compliance and commercialization concerns, even in basic science research. And Lorna, even though she's not recognized as a biomaterials person necessarily within our community, she's someone who her cellular solids work has provided a framework for us to understand how cells interact with porous materials in general. And last, I also want to thank my postdoctoral advisor, Les Silberstein, who invited me into a joint program for transfusion medicine, a combined clinical and research postdoctoral program where I got to be in the hospital and seeing how clinical treatments work in the blood bank, to not only think about what we're doing in the lab, but also think about what we can do in the future. All right, so I want to talk a little bit about the science that we've been up to over the last few years. And like most people who have been on the stage, there's some variation of this slide we've used where the idea has been the past and the future of tissue engineering. It still thinks about how do we combine biomaterials and cells and some sort of biologic. And we've been using this for the last 20 or 30 years as a paradigm to think about how do we replace, restore, regenerate tissues across the body. And some of the work that my lab has been up to, it falls very much in that realm, and I'll talk a bit about it. But I also have really appreciated over the last few years how the society and our members have really pushed forward this idea as well, is how do we use material systems and tissue engineering approaches to take things outside of the body and develop biomimetic platforms to understand mechanisms of cell bioactivity, or to think about clinical platforms or individualized medicine approaches. I think they're all really exciting. So a little bit of a background, and so I'll try to summarize my graduate experience in one slide. And essentially what we came up with was a very basic question my advisors got me thinking about, but it's one that has motivated me to this day. And it was a very simple question. We're working with porous collagen, glycosaminoglycan, or CG scaffolds, and we asked the question, as we change the pore size of these scaffolds, how does that impact cells within the material? A very basic question, you might imagine, but one in which that allowed me to build an understanding of the cellular microenvironment in 3 dimensions. And so there's a number of works that we've worked on over the years, but one thing I wanted to point out is you take these porous scaffolds, the pore sizes are larger than that of cells. As we change the size of the pores, numbers of things shift. Not only do the permeability of the material change, but also features such as the flexural rigidity of the individual fibers their cells are sitting on change. So even though we have a 3-dimensional material where we're changing the size of the pores, we can see differences in mechanotransduction signaling pathways, and we see significant effects of cells because of differences in surface, specific surface area, permeability, flexural rigidity, and the spacing of these junction points between fibers. And these all matter. But most importantly, this relatively simple question got us thinking about using the ideas of cellular solids approaches to model the cellular microenvironment. And so it's provided a really unique insight for us to take a step back in my lab and start thinking about new questions inspired by this initial work. And so my lab is generally motivated by the idea of instructive biomaterials. And this was not something we started at, but actually came as a partnership between me and my students. As projects developed, as new opportunities came, we've sort of had a series of projects that have developed over the years, and I'm excited to talk about them. But they all come down to this fundamental question. We have a cell, we have it in a material. How do we develop an instructive material to drive this response? And I won't be able to talk about all the work we've been up to, but generally we've been thinking about ways we can pattern microstructure, we can pattern biomolecular signals, we can deal with compositional properties, or even ways to regulate cell-cell communication, all in the service of thinking about how do we move from the idea of a uniform block of material to a material that has some minimal degree of complexity within it in order to address a particular clinical question. And so while the time is short, I won't be able to talk about everything. I do want to talk about 3 sub-projects that have been developing in my lab, and all of them are actually was well introduced by Dr. Zemsky's keynote on Wednesday, where he introduced this idea where we wanted to make sure that our work has translational value and we're thinking about where it goes. And so all these different efforts have some clinical translation in mind. How far we've gotten is different on each project. But it's really a unique opportunity to think about how do we motivate our work on what we see in the body. And so I'm going to talk about 3 projects. One has to do with engineering orthopedic insertions, more of a regenerative medicine project, and 2 others, a glioma on a chip and an artificial bone marrow platform, that are both in the realm of developing individualized medicine treatments. And so the first project we really got started in my lab was really dealing with an interesting question. So we looked at orthopedic insertions in the osteotendinous or tendon bone junction in general, and this junction connects highly elastic aligned tendon to mineralized bone. And it's not a hard transition. There's actually a gradient interfacial zone where you see overlapping patterns of matrix proteins, mineral content, growth factors that together provide a biological interface between 2 tissues that have over 2 orders of magnitude difference in stiffness. It's also a region where you see significant clinical failure, and the current methods, which are mainly mechanical fixation of tendon to bone, have very poor healing response in some classes of patients. So we got thinking about this as a motivating system. Both there's a clinical need to think about a way to regenerate this biological insertion, but also as a really interesting platform to develop material systems that deal with the spatial complexity of the orthopedic insertion. And so our goal was to try to develop a material that could be sitting on the shelf in an operating room, that itself had some sort of instructive parameters built into it, that you could harvest the patient's own mesenchymal stem cells or adipose-derived stem cells, seed them into the material, and put it back into the patient intraoperatively, and in a manner that the material itself provided spatially selective instructive cues to drive differentiation. A lofty goal, but one that we think it really motivates the development of some really interesting biomaterial science. And so the questions we developed, we addressed early on were ones that were really inspired by the great work over the last 2 decades, looking at how cells respond to mechanical cues in 2-dimensional surfaces, and thinking about, as we abstract this into 3-dimensional materials, how we can get similar types of responses. And so the basic question we had is, as we transition from this isotropic, porous collagen biomaterial to one that is highly anisotropic or one that is highly mineralized, how we can use that as a way to drive selective differentiation of stem cells. And so this is what one of these porous collagen gag scaffolds would look like in an isotropic form, and over the years we've developed some directional solidification methods so that we fabricate these via lyophilization to create highly anisotropic scaffolds with ellipsoidal pores that are stacked together, or other variants that have a calcium phosphate nanocrystallite built into the material that not only stiffens the material, but also some of the calcium phosphate content is released over time, potentially providing a bioactive signal to drive this response as well. So the question was a simple one. Instead of using the large doses of growth factors that we might be able to use to drive osteogenic differentiation or the like, are there ways that we can use the structural parameters of our material to drive similar types of responses? Can we drive a pre-tenocyte phenotype simply using the degree of anisotropy of the material? And can we drive a pre-osteoblast phenotype using the mineral content of the material? And this came out of nowhere. It actually came out of a series of studies done early on where we were isolating primary equine tenocytes and looking at their bioactivity inside of materials, where we noticed as we transition from these isotropic to anisotropic aligned materials, we saw significant cell alignment. So the structural properties of the scaffold are highly aligned, so the focal adhesion sites on the cells were becoming highly aligned. And we found in equine tenocytes that these cells would remain primarily aligned in the direction of scaffold anisotropy. We confirmed this using a cardiomyocyte study as well to look at a different tissue where you similarly see highly anisotropic tissue structures. But most interestingly is when we designed a series of these anisotropic materials that could be structurally remodeled over time. So as the cells were able to contract them, we saw a loss of all the proteinogenic gene expression profiles with cell contraction and loss of anisotropy. So it gave us a clue that this anisotropy might be important. And so we then embarked on a series of studies where we looked at taking mesenchymal stem cells, human bone marrow-derived mesenchymal stem cells, seeding them in these materials, and instead of using a selective media like an osteogenic media or the like, we simply had them in growth media and asked questions about how the structural parameters of the biomaterial would impact cell response. Through a series of work by a number of students in my lab, we were able to show that the degree of anisotropy of the scaffold sufficient to drive increases of markers of early tenogenic differentiation, such as scleraxis, in the absence of even mechanical loading. And on the other side, the incorporation of the correct mineral content, not at a full 80 weight percent you see in bone, but at a lower weight percent, we could drive selective upregulation of osteogenic differentiation and matrix synthesis in the absence of soluble factor supplementation. I want to just highlight one poster from Dan Weisgerber in my group, number 489. He's got some of the more in-depth development of this mineralized version of the scaffold. But we were still dealing with two materials here, where we had a highly aligned and a mineralized version, and we so wanted to ask questions about how we were able to create an integrated biomaterial structure. And for this, we had our first turn down an interesting motivation road, thinking about the development of a black and tan or a half and half, depending on whether you're from England or Ireland. And the idea here is that you can layer alcohols, and if you let it sit on the bar for long enough, you get interdiffusion zones that will form between the layers of alcohols. So we use that as a motivation, where we started to create our mineralized and our non-mineralized collagen slurries, allow them to interdiffuse, and then freeze dry them. And what we're left with is a single biomaterial where we have a highly aligned non-mineralized side and a mineralized side, and most interestingly, an interfacial zone that's not a discrete interface, but a gradient zone itself on the order of about 250 microns, which is on the size and scale that you see of the native osteotendinous junction. Most importantly, though, is we could take the same mesenchymal stem cells and seed them in our biomaterial, in this case putting it on a modified flex cell bioreactor in the absence of any sort of biomolecular stimulation. We're doing this solely with growth media. We could take these mesenchymal stem cells and load them in these materials and see preferential osteogenic differentiation on the bone side, preferential tenogenic differentiation with additional stretch on the tendon side. And so it's really started to think about a material, one cell source, instructive material, spatially selective response in the absence of biomolecular supplementation. It's also led to lots of really interesting questions. And a project one of my students, Laura Mosden, who had a talk yesterday, started to look at as we change the diffusion times, we may have the potential to change the width of the interface and hence the intensity of the gradient of structural mineral properties these mesenchymal stem cells are experiencing at the interface. So there's a lot of really interesting projects we have under development now. We're starting to ask more questions about how we can play with the mechanobiology of the interface to drive cell response. I want to give a little bit of mention to more traditional supplementation methods. My lab has not been adverse to that. In fact, we've had a series of publications over the last few years where we've really started to think about the use of composite media supplementations to enhance proliferation and enhance phenotype. We've also started to look at the dangers of using mixed media supplementation strategies where the biophysical parameters of the material itself could drive unexpected differentiation results in mesenchymal stem cells. We've had a series of work also looking at ways to translate this to a more spatially selective manner, where some work looking at ubiquitous incorporation, and as Bill had mentioned, a benzophenone project using selective immobilization of growth factors, where we're not only looking at immobilization, but potentially also release of these factors over time. And lastly, some really fascinating work that a student, Rebecca Hortensius, in my lab has been working on, where she's been looking at using, inspired by Bill's lab, proteoglycans in our biomaterials to transiently sequester growth factors, to intensify a response or provide some selective representation of cell response. But one of the things we've noticed in doing this is that it's very hard to do these studies. We start with biomaterials, we start with our cells, and the next thing you know, you have literally hundreds of materials that you're putting into culture and trying to maintain. And so had a really industrious student, a master's student, Emily Gonerman, who had started working on an array-based biomaterial, where it's an array scaffold we still fabricate with freeze drying, but it gives us a platform to do in a 96-well plate format orthogonal gradients of microstructure and biomolecular patterns. And it's allowed us to ask multiple questions. So if we have overlapping patterns of PDGF and BMP2 immersed within this biomaterial, there are ways we can selectively drive proliferation or osteogenic differentiation and the like. And so it's giving us a proving ground where we can create an array chip, read the results out in a plate rater, and put the array chip back in culture in order to do this sort of in a high throughput manner. One other project I want to touch on just briefly in the idea of biomolecular supplementation is that one of the big problems we all know we have is driving new vascular formation. And my lab has been cognizant of it for quite a while, and it took a student to really take a risk on a project. And so I want to just highlight some really exciting work that Jackie Pence in my lab is working on right now. So how do we build vascular structures into materials, and how do we provide some orthogonal cue in order to do that if we're already worrying about spatially selective properties themselves? So she took a page out of a completely different system, looking at the endometrium, the lining of the uterus, which is a really interesting tissue because of the speed by which you get rapid vascularization over the course of the menstrual cycle, but also rapid vascular reorganization right after embryo implantation. And so it's a highly organized process that is not motivated explicitly by high doses of VEGF, and so we thought that was quite exciting. So she's taken on this project where we were looking at the cellular crosstalk between endometrial epithelial cells and endothelial cells and ways we can use biomolecular signals such as estrogen and progesterones to drive a provascular response. And so I just want to highlight some work that she has at a poster downstairs, number 491 as well. It's sort of thinking about new strategies of ways we can approach the vascular revascularization issues that also consider things such as the changes in estrogen and progesterone that come with endometrial cancers. So perhaps some of the most fun we had started as a project in my lab for an undergraduate. And it's a problem that we're all also faced with, is that the idea that we need to have materials that are structurally competent and bioactive. And in porous scaffolds, that is really the main player, especially working in orthopedics, is that there's this trade-off between scaffold bioactivity and mechanics, is that you can make the scaffold strong by making it less porous or more dense. The problem then is you have biotransport concerns and a loss of cells. And so we thought about how is this problem solved in other fields of venue, and we looked to nature again. And here we got inspired by core shell composites. So this is cross-sections you see of a porcupine quill on the left and a sunflower stalk on the right. And so you think about the sunflowers we have out in the prairie, they're way high up in the air. You've got these giant stalk structures that support the weight of the sunflower but allow diffusive transport. And the way this happens, you have a high density shell that gives strength to the composite in bending and tension and torsion, and then you have a porous core that provides the bioactivity. And so it started as an undergraduate project of Manny Ramirez, who's now a grad student at the University of Rochester, thinking about are there ways we can create collagen membranes, high-density collagen membranes. And so we came up with an evaporative process to create these high-density membranes, and using a version of our black and tan fabrication approaches, we actually can create these core shell scaffolds. So on the bottom left there is one of our core shell scaffolds, a high-density core, a high-density shell and a porous core, and most importantly, by changing the shell thickness, we can improve the mechanics of these scaffolds. So we can now get a 99% porous scaffold core having an elastic modulus of the hydrated material approaching 10 megapascals. And so that's a really nice advance. And more recently, some work of Stephen Cagliari was starting to look at ways that we can make these scaffolds periodically perforated. So the membrane is perforated to allow better cell perfusion in and better cell bioactivity. And lastly, some work in the bio-inspired realm of Laura Mosden, where we started to get inspired by the interdigitations we actually see at the tendon-bone junction and a series of other interfaces across the body, where not only having an increase in the surface area by having these interdigitated structures improve strength, but also changes the mode of mechanical loading. And so she asked a very basic question: if I could create a series of these interfaces between mineralized and non-mineralized scaffolds that were either flat or had degrees of interdigitation built in them, is there a mechanical advantage? And the preliminary results suggest we see increases not only in the modulus in the material, but also increase in the failure mode by going to these interdigitated structures. So I want to shift gears slightly in the last few minutes that I have and start thinking about other approaches that we can use for biomaterials. And so I'm going to talk about an ex vivo platform, engineering the hematopoietic stem cell niche. So for those of you who are familiar with hematopoietic stem cells, you and I all have them in our marrow. They're responsible for producing our entire complement of blood and immune cells, almost 1 trillion cells a day. But there's a number of things that can go wrong within the marrow, and I want to talk about one that we really got inspired by. So I could tell you that the inspiration for this project came out of my experience as a postdoc, sitting in a blood bank and seeing the process of hematopoietic stem cell transplantations that were used to treat leukemias, but I'd be leaving out an important part of the story. And so on the right there is actually a picture of me when I was 17. I was diagnosed with leukemia, and it really motivated a lot of what the work is going in the future. And so I was treated with a bone marrow transplant. That's actually me as I was receiving my transplant. It's not something fancy. They take a bag of your donor's marrow, they hang it on an IV arm, and they go and they let the marrow diffuse into your blood. And now these hematopoietic stem cells within the transplant are expected to replace your own marrow that has been ablated due to radiation and chemo that you had to have in order to treat your leukemia. There's a number of problems associated with this, and this is what motivated us to really think about engineering solutions for a hematopoietic stem cell niche. And so if you look at the survival rates, and these are current day survival rates for these patients that are treated by hematopoietic stem cell transplant, even though it provides in some cases the only option for treatments of a range of these diseases, at best case you're still in the order of 50% survival out to 5 years just from the transplant itself. And that's for if you're relatively healthy, other than the fact you have leukemia. If you don't have full remission or you're older, these numbers get a lot worse. And the question is why? And this first year is really critical post-transplant. And there's a number of complications that come up, some that we're not dealing with, but some that we are. And the two that I wanted to highlight is this idea of low homing efficiency and engraftment. So if you recall, I said they give you this bag of marrow, they hang it on an IV arm, and they put it into your bloodstream. These hematopoietic stem cells are expected to home back to your marrow cavity, home into the cavity itself, and then engraft, set up a functional hematopoietic marrow. And those two processes have very low efficiencies with transplant. And so we saw that as an opportunity for engineering tools to both understand the process of homing engraftment and come up with platforms in order to improve clinical therapy. So examining the marrow, what we were struck by is it's not just a giant ball of fat. There's a lot of interesting structural and mechanical and biomolecular properties across the marrow that have been characterized over the last 20 or 30 years. We know quite a bit about this process, about how these hematopoietic stem cells are homing from the blood vessels through the marrow, where they're exposed to a series of different marrow architectures and environments. You have gradients in extracellular matrix proteins such as laminin and collagen and fibronectin, gradients in biomolecular cues, and also known gradients of where the more quiescent or more active hematopoietic stem cells lodge. And so we saw this as a unique opportunity to ask questions about how do we engineer material systems inspired by the native niche that could drive either selective expansion of a long-term repopulating hematopoietic stem cell to solve this engraftment problem, but also understand more about the process by which these cells home and engraft within the marrow itself. It's a process that's too rare to track in the body because these cells are so rare, but it's one that you can try to develop material tools. So along the way, we started thinking about how might we really want to engineer the stem cell niche. So you think about the trajectory our our field is on over the last few years. The last 20 years or so of really starting to think about the stem cell microenvironment has brought us a whole host of advancements, starting from combinations of biomolecular cues in 2D culture, where you're looking at the time of presentation, the factors that drive differentiation, starting to think about how do you build this into a three-dimensional architecture. You have the opportunity to not only put in place structural and mechanical cues, but also limit the diffusion or use the backbone of your three-dimensional materials to provide instructive biomolecular signals tethered within the matrix itself. But also you have the opportunity to ask some really interesting questions about stem cell niche cell interactions. And so this really motivated our work, where there's been some pioneering work in our society from Chris Roy and the like, started to ask questions about presenting cadherins and other cell-cell molecules on the surface of microparticles and seeing whether or not you could use that to engineer hematopoietic response. And so we said, not only do we have to worry about these cadherin type interactions, but the niche is a three-dimensional environment. We also have to think about balance of paracrine and autocrine signals that might be taking place within the niche as well as the remodeling processes. And so we need a biomaterial framework to investigate this. And so some work over the last 6 years, started with Sunny Choi in my lab. She asked some very basic questions about the response of hematopoietic stem cells to matrix mechanics and matrix environment, and looking at combinations of extracellular matrix proteins and elastic moduli that are inspired by what we see within the native marrow. She saw she was able to change the proliferative index of these cells as a function of matrix environment, but most notably she was able to see selective upregulation of maintenance of an early stem fraction through combinations of not only presenting fibronectin and a matrix-relevant stiffness of the biomaterial surface. She's since gone on to show that she could selectively abrogate this response both by blocking mechanotransduction pathways, but also blocking integrin-ligand interactions. She's also seen that in order to drive downstream differentiation, she could also use matrix signals, and using matrix cues inspired by regions of the marrow where you see more active cycling of hematopoietic stem cells, we can drive downstream differentiation of these cells as well. I had a separate student, Bhushan Madik, who started asking a really interesting question. The marrow is multicellular. We've got a hematopoietic stem cell surrounded by a range of niche cells. What happens when we start putting these together in controlled environments? In 2D culture, we could take our hematopoietic stem cells and a niche cell taken from the active marrow that's known to drive proliferation and differentiation of these HSCs, and show by putting them together in liquid culture, you can get selective upregulation of proliferative index of these hematopoietic stem cells, and you could do so in a dose-dependent manner. The more these niche cells provided, the more we drove this proliferative response. But the more interesting question you started to ask is, if we introduce a biomaterial matrix within this environment that can regulate the diffusion of small molecules, are there ways that we can shape this response in more interesting fashions? And so, inspired by some of the work that Peter Zanstra has been doing, looking at autocatalytic responses in hematopoietic stem cells, we created a system where, I have to apologize some of the busyness on the bottom, but if you look at the bottom right, we've got a 2x3 matrix, and the way to read this matrix is going in the rows from the bottom to the top are hematopoietic stem cells combined with more and more niche cells, so increasing paracrine load. And as you go in the columns from the left to the right, that's increasing the density and decreasing the diffusion rate of these gels. So that of these 6 regions, the results you're going to see on the next slide on the bottom right would be most driven by autocrine type responses, and the results you see on the top left would be most driven by paracrine type responses. And so what he noticed was quite interesting. If we take these hematopoietic stem cells, mix them with these active niche cells that are known to drive differentiation, what we see is, if we look at the early progenitors, we see a selective increase in these early progenitors in the environments where we can actually have increased autocrine type signals, a material that has no niche cells and it has a much lower diffusivity of small molecules. We can enhance sort of autocrine responses to drive maintenance and expansion of the early stem fraction. And if we look further down the differentiation pathway, what we're able to see is that in the gels that have the highest degree of paracrine type signals, we drive the opposite. We can drive downstream differentiation, represented by the darker colors here, as a selective increase with blue versus red relative to 2D controls. And in fact, he started to map this out as we go down the full hematopoietic stem cell differentiation process, and it's held true, is that autocrine responses seem to be driving a lot of these early stem responses, and these paracrine factors are driving some of these downstream differentiation factors. And so it's really opening up a new way to think about how do we engineer the stem cell niche. That's quite exciting going forward. In the last 5 minutes or so, I want to talk about one last project. There was an offshoot of a project, and it was one that was really driven by an interesting question, driven by an excellent postdoc, who asked questions about, can we do better? And so this project is really motivated by treatments for glioblastoma, which is a particularly common and lethal form of brain cancer. As Dr. Belancanda introduced yesterday in his keynote, has a very poor median survival, and these survival rates are essentially unchanged over the last many decades. And the reason for this is that it's an incredibly difficult disease to treat. We have diffuse spreading of these tumors, you have undefined tumor margins, and an inability to do anything other than provide palliation. And so we started asking questions, are there ways we can have biomimetic cancer platforms to look at new ways to treat these tumors outside of the body? And so the inspiration came from talking to our colleagues up at Mayo Clinic and understanding how they go through the development process of small molecules to treat these diseases. The xenograft model has been a workhorse in the cancer community over the last many decades, where you take patient biopsy cells, you inoculate them into colonies of mice, and generate these orthotopic tumor models where they're targeted to the tumor itself. You have different colonies that are targeted to each patient you're going to be dealing with. These are expanded many times over, and it gives you a framework in which you can test combinations of therapies. It's an incredibly time-consuming, incredibly expensive process, and it's one in which we started asking questions, are there ways that we can develop high-throughput or combinatorial biomaterial systems to cut down on the type of xenograft models we see here? And so are there ways we can replace serially propagated xenografts is one of the big inspirations we have to maintain patient tumor morphologic and molecular characteristics to test new combination therapies. But more excitingly, this idea, are there ways that the knowledge we gain from that type of work, how can it translate into the clinic to provide a platform to do real-time readjustment of clinical therapies on patient samples? The idea that you could take a biopsy out and have it run in parallel with the patient as you test combination therapies on the chip before adjusting the patient's dose is quite exciting. So to do that, we needed a material system and we needed an approach. And so Sarah's work over the last few years has really concentrated on developing a methacrylated gelatin platform to do this in. The reason we chose this is that it gave us an opportunity to not only tailor things such as the structure mechanics through changing the weight percent and degree of functionalization of methacrylates, it also gave us an opportunity to graft in biomolecularly relevant properties, such as in this case, adding in hyaluronic acid into the gel to mimic the native brain matrix. We found we could selectively define the right structural, mechanical, and amount of hyaluronic acid we tuned in to tune the malignancy of these glioblastoma cells, effectively thinking about ways to create a biomaterial rheostat. But what I want to end on is this idea that that's not maybe good enough, and that was really the motivation over the last few years, is that we could create a jello block that has these tumor cell lines sitting inside of it. We could look at relevant responses, but this is what a tumor actually looks like. If you go talk to the neurosurgeons, what are they dealing with is it's an undefined tumor that is spread throughout the brain, and you have the tumor mass that itself is not a uniform block. You've got overlapping gradients of matrix proteins, hypoxia, mechanical features, structural cues. Then you get to the tumor margins, and as Dr. Bellamcana really said very well yesterday, the neurosurgeons say, you know, I can't take everything out. You know, where do I end? How do I know how far to go? And what about the graded properties right at these tumor margins can be used to predict the patient survival long term? And so for that, we needed a new approach. And so we were playing with these microfluidic mixture tools. There's one that we all have used in the past to create gradients and growth factors. We decided to apply it to hydrogel systems. So our version here is we have two inlets of our hydrogel precursor suspensions that could have any combination of cells or matrix or growth factors, runs through the mixer and gets spit out into a mold. And this mold can vary in size from as small as 20 microliters up to a few hundred microliters. And so the resulting hydrogel is three-dimensional. It's about 500 microns thick, and it has overlapping patterns of whatever went through the microfluidic mixer. So we've shown we can create overlapping patterns of cells. Had a really industrious graduate student, Bhushan Mata, who developed a way to selectively remove parts of this gel over time to look at spatial responses and be able to pull out what he called a marrow zip code, or a tumor zip code, where we could look at a selective region of the gel to do downstream processing such as looking at flow cytometry or gene expression or secretome type data. And because they're so small, we can actually look at individual cells within the gel as well. And so we've translated this into the marrow, and I won't be, from the marrow into now thinking about the brain tumor project. And I won't be able to talk about all the results, but just one was a single hydrogel where we can create gradients of cross-linking density across it and use this to be able to show selective upregulation of cell proliferation and changing gene expression profiles to more malignant phenotype across the gel, but also being able to measure secretome information, in this case, production of MMPs, because we're doing it in a highly controlled three-dimensional environment. So it gives us this opportunity to start engineering spatial responses across a single material. And we've demonstrated this for creating, in this case, structural gradients. We've also shown it for cell density gradients and matrix protein gradients as well. But lastly, the challenge is taking it to the clinic. Everything works very well when you use cancer stem cells in a very controlled environment. You can make them do whatever you want in a reproducible fashion. The trick is when you go to these patient biopsies. And glioma is particularly interesting because it has such a range of genomic mutations associated with it. So we've been starting to work with Mayo Clinic, getting a series of these xenograph type models. In this case, I'm just showing you 4 of them that have increasing infiltration capacity, and have differential response to radiation, have differential response to a series of diseases. And this is the true clinical data set we see. And what we see is when we put them in our gelatin hydrogels, we get even a huge range of just cell proliferation. Whereas if we start to titrate in the right amount of hyaluronic acid, we might be able to start getting more consensus response between a whole series of different materials that are everything from non-invasive to highly invasive. And in fact, what we've also noticed is that depending on whether they're highly invasive or non-invasive or mildly invasive, by tuning the amount of HA within these gels, we might be able to selectively find the sweet spot for what's the real malignant profile of these cells. And so again, it's an opportunity to think about personalized approaches. So what I want to end on is actually my last slide. And what I want to talk about is something that actually Dr. Edelman went over very well yesterday. And so I won't go too much into it, but really emphasizing the need to invest in biomedicine, invest in researchers. And so what I have here is the timeline for the hematopoietic stem cell transplant that saved my life. It started in the early 1950s, some pioneering work of Jacobson and Lorenz, just noticing that you could protect the spleen of mice and they could survive radiation therapy better. And that translated into the pioneering work of Don Thomas, who won the Nobel Prize for this in the 1970s. As early as the mid-1950s, he was experimenting with this idea of using transplanted marrow cells in order to rescue mice first and then patients after high doses of radiation and chemotherapy. The first transplant didn't even happen until the late 60s. The first transplant in a patient with lymphoma or leukemia didn't happen to the mid-70s. And by 2007, we've got almost 50,000 patients a year being treated with this therapy. And so that's the life cycle of these treatments. I think Dr. Edelman went into it in real detail, but that's really what we need to be focusing on here. And there's a danger in our scientific community right now, and it would be irresponsible for me not to talk about it, is that it's important for us as a society and important for us senior, junior trainees to think about how do we convince the public and how do we convince people of the value of our work. And I think it's incredibly important thinking about how do we balance basic science and applied research endeavors. It took 50 years to develop the therapy that is saving 50,000 people a year. The same types of timelines are in place for so many of the projects we all work on. And with the changes in how we've done things such as FDA regulations and funding rates, particularly for young investigators, there's a crisis that's been brewing for a long time, and the impact will be felt decades into the future. So I just wanted to end with 2 thoughts, is that I really have been inspired by the work of our society and the work of the people here, thinking about how do we better engage the government, how do we better engage the public, how do you explain to your grandmother why what I'm doing now with the $10 she gave to the American Cancer Society is going to benefit someone 15 years from now, and how do you invest in that? And it's also important for us to think about better and new and novel ways to form mentoring relationships. I've been inspired by so many relationships I've had in this society. Myself as a junior faculty with other senior members who took an interest, other junior members, if nothing else, who went and talked about pay lines and the rest. But these relationships are critical for building our society and building the strength of our research endeavor going forward, and thinking about how do we bring in the next generation? How do we convince that second-year grad student that this is the best job in the world, and this is a job that you can do well, and that we have your back and are providing an environment in which you can make a difference in a decade or 2. And so with that, I want to thank you for the opportunity to talk about some of the really exciting work that my group has been doing. It would be nothing without the hard work of my students. And I want to end with thanking my trainees again, the alumni from my lab, my collaborators who are too many to even list except in small font, all my colleagues who make this possible, and the funding that, the right to the checks. And so with that, I want to thank you again for this opportunity in the society.